So the first step of viral replication is the attachment. So this is the so here the surface proteins of the virus. So they interact with the specific receptors on the target cell surface, and these may be specialized proteins with the limited distribution, or the molecules that are more widely distributed on the tissues throughout the body. And the presence of the virus-specific receptors is necessary, but not sufficient for the virus to infect the cells and complete the replicative cycle. So the very important thing that, as I told you, so virus should have an attraction towards the certain host cell. So without that one, so they cannot attach, or they cannot multiply. So usually in case of the in case of the influenza virus, so the hemagglutinin which is present on the virus surface attached to the glycoprotein receptor site on the surface of the respiratory epithelium. And in case of the polio virus, so the receptor is the lipoprotein which is present on the surface of the primate, and but not in the rodent cell. So that is the reason. So the polio virus cannot attach to the, cannot infect the rodents. It can infect only the primates. So here you need to know, so understand that that is the attraction between the receptor. That is a lock and key mechanism. So only when there is a appropriate key for that lock, so then only there will be an attachment of the virus to the host cell. So this the first the takes place in two steps. So first there will be a preliminary attachment by the ionic bonds and the charges, and it can be reversible. But second, more it will be more specific attachment to the certain receptor sites, which is more firm and irreversible in nature. So now that is the first step will take place, that is the attachment of the virus to the host cell. And next one is the penetration. So now the virus has to enter inside the host cell. Once it is attached, so it has to enter inside the host cell. And the penetration takes place by any one of the two methods. So it is the first one is called as the viropexis, where the whole virus is engulfed by the cells in the phagocytic process. So that means it's just like the phagocytosis. So the virus will be completely engulfed by the host cell. And the another mechanism is in case of the enveloped virus, where the viral lipoproteins which are enveloped, they fuse with the host cell surface membrane, and it is called the fusion method. And this results in the release of the viral nucleocapsid material into the cytoplasm of the host cell. So in any of the two methods, so the host cell or the virus, it will enter inside the host cell. So in this one, you can see that the first one is the endocytosis. It's just similar to that of the phagocytosis. So once there is a specific attachment on the surface of the host cell, so this attachment, it starts certain biochemical reactions, and because of which the host cell, it will just engulf the whole virus inside. And once the virus is inside the host cell, then there are certain enzymes like the lysozyme enzymes which are present in the host cell. So these enzymes, so they will dissolve the outer protein coat. And once the outer protein coat is dissolved, and then the free DNA or the RNA, it will be released out into the host cell. So this is through the endocytosis process. And the second one is the fusion method. This will happen in case of the enveloped virus. So when there is enveloped virus, so once again, because of the specific attachment site, this is the receptor site on the host cell, and these are the enzymes which are present on the virus. So there is an attachment, and once the attachment takes place, so this initiates a biochemical reaction, so which will fuse the outer membrane, that lipid membrane of the virus, with that of the membrane of the host cell. So because of that, yes, Vishaka, any question? Anybody has got any question here? Any questions?
No, sir.
Okay, somebody has raised the hand. That's why I'm just asking. If you have any question, you can ask me. No problem in between. Vishaka, she has raised the hand. I don't know. She's having any doubt. Anyway, let me continue. So because of that fusion of the viral outer membrane, that lipid membrane with that of the host cell, so that will release the nucleocapsid inside the host cell, and then once again the enzymes which are present in the host cell, they will uh, dissolve the outer that's capsid layer of the protein layer of the virus, and the genetic material will be released into the host cell. And in some cases, that's just like in case of the uh, bacteriophage virus. So there is a direct injection of the nucleic acid, of the genetic material into the host cell. So that is called as the nucleic acid translocation. So during this nucleic acid translocation, there will be an attachment, and after the attachment, directly the virus, it will inject the genetic material into the host cell. So there's very few cases occurs like that, but otherwise, usually the second step, that is entry, it takes place by either by endocytosis or by the fusion method. And next is the uncoating. So third step is the uncoating or disassembly. So here, so virus is stripped off its outer protein coat, and that is and the capsid, so that the nucleic acid is released into the cell, and most virus, their uncoating is offered by the action of the lysozyme enzymes in the host cell. So usually the lysozyme enzymes which are present in the host cell, they are having the ability to dissociate or to metabolize the proteins and they dissolve the protein. So what they do is they just attack the protein coat of the virus and making it easy for the virus to release genetic material. If our host cell doesn't do that one, then viral infection actually doesn't take place. But it is the nature, it's the part and parcel of the lysozyme enzyme, so which will do that one. So that is the viral second, that is the third step, that is the uncoating, or it's called as the disassembly. And the very important step, that is the fourth step, that is the biosynthesis. So this biosynthesis, it takes place by translation, transcription and translation. So in order to understand this one, if you should be understanding so how the protein synthesis will take place. So during the protein synthesis, so first, as I told you, the DNA, it will open up a segment of the DNA for which is responsible for synthesizing that particular protein. It will open up and it will synthesize an complementary copy, which is called as the messenger RNA. So this messenger RNA takes up the message which protein has to be synthesized, and that messenger RNA comes out of the cytoplasm, and it, with the help of the ribosomal RNA and the transfer RNA, it will go on adding the protein, the amino acid particles, and it synthesizes the protein. So here, in case of the virus, so the viral genetic material, this will take the lead, and this viral genetic material directs the host cell to synthesize the viral proteins. So this is what happens. This takes place in two steps, that is the transcription and the translation. So this immediately after the penetration, and an interval of time is called as the latent period will start in case of the viral infection, and during which the uncoating of the virions will take place, and some virus, they are uncoated in the cytoplasm and some will be uncoated in the nucleus. So here, during this stage of called as the latent period, the viral nucleic acid, the capsid proteins, the enzyme necessary for various stages of the viral synthesis, the assembly and release, and they are all produced. So during this stage, one cannot investigate the presence of the virus inside the body. So because the virus, there will not be a virus present, so it will be the part of the virus which will be present in the cell. So that's why it is called as the latent period. So where we cannot be able to identify or diagnose the presence of the virus. So during that stage, so first usually the viral genetic material, it produces a regulatory protein. So these regulatory proteins, they shut down the normal functioning of the host cell, and then it will direct the host cell to synthesize the viral particles or the viral components. So the site of viral synthesis, this depends on the nature of the virus. most of the DNA virus, this viral synthesis takes place in the nucleic acid, in the nucleus, and whereas for the RNA virus, it takes place in the cytoplasm. But for some exceptions, some DNA virus the replication of the synthesis may take place in the the cytoplasm, and for one RNA virus, it will take place in the nucleus. So there are some exemptions. So otherwise, so the DNA virus replication takes place in the nucleus, and RNA virus replication takes place in the cytoplasm. And the biosynthesis takes place in the 4 different steps. In the first step, there is a transcription of the messenger RNA from the viral nucleic acid. So if it is a DNA, then it will synthesize the messenger RNA, and that messenger RNA will take the further steps. And if it is a RNA virus, so that RNA itself, it acts as a messenger RNA. And if it is a negative sense RNA, then that negative sense RNA, it produces a complementary copy of RNA, and that complementary copy, it acts as a messenger RNA. So the first step is the transcription of the messenger RNA. And then the translation takes place, second step, that is the translation of messenger RNA into the regulatory protein. So that is the second step. And the third step is the replication of the viral nucleic acid. So now once the messenger RNA is synthesized, so that will help in the production of the viral proteins, and but at the same time, in the third step, so there will be a replication of the genetic material. So for the daughter variants, it requires the genetic material. So then there will be a generation of the genetic material. So if it is DNA replication will take place, and if it's RNA replication will take place. And then in the fourth step, there is a synthesis of the late or structural proteins, so which are components of the daughter virion capsid. So these proteins, that is the structural proteins, will be synthesized. So this synthesis will take place in 4 steps: the transcription, translation, the replication of the viral nucleic acid, and the synthesis of the late or the structural proteins. And in case of the poliovirus and other single-stranded RNA virus, so the viral RNA acts directly as a messenger RNA. And in the parental RNA, that will be, which is a positive strand, act as a template for the production of the complementary strand, that is the negative strand, and which in turn acts as a template for progeny of the viral RNA. So in some other single-stranded RNA virus, like influenza virus, so the parental RNA, it produces the negative strand, which acts both as a messenger RNA and a template for synthesis of the progeny RNA. And in some virus, so where RNA and DNA hybrid is formed, so ultimately they produce the viral DNA, that is a double-stranded DNA, and which gets integrated into the host cell genome. And this acts as a template for synthesis of the viral RNA. So this is how in the different virus, so there is a different mechanism for the genetic material, replication of the genetic material. So depending on the need or depending on the nature of the virus, so this multiplication will take place. So then the next step is the maturation or the assembly and release. So after now the synthesis, the next step is the assembly, and it may take place inside the nucleus or the cytoplasm of the host cell. And usually in case of the herpes and adenovirus, assembly takes place in the nucleus, whereas in case of the picornavirus and the pox virus, so it takes place in the cytoplasm. And here non-enveloped virus, they present intracellularly as a fully developed virions, whereas in case of the enveloped virus, only the nucleic capsid is complete, and the envelope is derived from the host cell membrane during the budding. So once again here, the non-enveloped and enveloped virus, so the things are quite different. If it is a non-enveloped virus, so inside the cell only, the daughter virions, the fully matured virions will be ready, and they come out of the host cell either by using cell lysis or by any other mechanism. But whereas in case of the enveloped virus, so inside the host cell only the nucleic capsid is ready, so the envelope is not yet ready. So that will be taken up by the virus from when it is budding out of the host cell. So this is in case of the normal non-enveloped virus. So here the entry of the virus and the multiplication, the replication will take place. So these daughter virions which are present inside, so they just come out of the host cell by breaking it down or by the cell lysis. And in case of the enveloped virus, you can see here, so this is the RNA in just example, and the RNA, then the protein coat will be produced, and this protein coat will be attached at the bottom of the host cell nuclear membrane, and then while coming out of the host cell, it acquires the protein coat as well as the host cell membrane, that is the envelope. So this is how the maturation and the release of the virus will takes place. And the last is the release, as I told you, in case of the bacterial virus. So the release of the progeny virus that takes place by the cell lysis. And in case of the animal virus, the release occurs without the cell lysis. And these progeny virus, they are released into the surrounding medium, may infect the other cells. And in case of the varicella virus, so this the daughter virus or the virions, the transmission occurs directly from cell to cell. So they don't come out of the cell into the external environment. In fact, they're just from one cell to another cell. The daughter virions, they just migrate. And the poliovirus, it causes damage to the host cell by the cell lysis. So that's why it causes the paralysis. And from the stage of penetration till the appearance of the mature daughter virions, so the virus cannot be seen inside the host cell. So because there will not be any virus, so this genetic material and the protein particles will be separated. So that's why we cannot just see the virus inside. In this one here, you can see this. So we cannot just see the virus here. So once it enters the inside, so the genetic material is there, the replication and protein synthesis is taking place, but we cannot see any virus over here. But only when there is an assembly and release, then only we can see the virus. So this stage is called as the eclipse phase. So where we cannot see the presence of the virus. And this time varies from virus to virus. And for the bacterial virus, it may be 15 to 30 minutes. And for the animal virus, it may be 15 to 30 hours. And in some cases, that latent period or the incubation period may be lasting for years. So sometimes the virus will be entering the host cell, but it will not cause any harm, or it will not start the transcription and translation process. So it may take up to many years. And even the same thing happens in case of the HIV virus. So HIV virus, once it enters the host cell, so it will become latent. It will be there in the latent condition for quite a long time in some people. And in some people, immediately it may show the response. So this is how the viral replication will take place. But still, since there will be one problem with the viral replication, so which is called as the abnormal replicative cycle. So this was been found by one of the Danish scientist, virologist, that is the Proben van Magnus. In the year 1944, he has observed this particular characteristic in the influenza virus. So it is called as the Van Magnus phenomena. So in the Van Magnus phenomena, what happens is that the hemagglutinin titer, it will be more, but the infectivity will be less. So that means we can see the antigens, that is the viral antigens will be present in very high concentration in the body, but the infectivity will be very less. So there are no symptoms and no infectivity. And this is due to the production of a non-infective progeny. And this is due to the defective assembly, or also known as the incomplete virus. So during the process of viral replication, the replication will take place, but during the assembly, so what happens is that the assembling, there's some fault will take place. There will be an improper assembly will be taking place, and which will cause the incomplete virus. So here the virus, it infects the host cell, and viral components are synthesized, but due to the defective assembly, either the release will be inhibited or by progeny becomes non-infective. So it will not be able to infect the other cells. And this is known as an abortive infection, or it's also called as the Van Magnus phenomenon. So this is the viral replication. So from the next class onwards, we'll go back to the bacteria, that is the growth of bacterial growth and factors affecting the growth of the bacteria. So with this, I'm going to stop the class, and we'll see in the next class.
Sir?
Yes.
Sir, could you share your slide notes, sir, yesterday's and today's?
No, yeah, that's what it's all there in the single slide. That's why I'm sending it today. Okay.
Okay, sir. Okay.